Okay, our next subtopic is method of estimation of lipids. So first part of methods. First of all, we'll discuss sample preparation, how we will prepare sample for estimation of lipids. It involves different steps. First step is drying of sample. In this step, water is removed from the sample. Main purpose is, we know that water is, inorganic solvent, and that's why it has to be removed from the sample so that it does not interfere in the sample preparation. So water is removed, and next step is particle size reduction. The larger size sample is broken down into smaller size, into smaller particles. Main purpose is that its surface area is increased so that its solubility is enhanced, so that it becomes soluble in some organic solvents in a better way. Next step is acid hydrolysis. As the name indicates, lipids are removed by heating in some acid and in some solvent, in proper solvent. Which type of solvent are selected? Solvent is selected. Lipids, we all know these are soluble in organic solvent, so that's why solvent which are selected, they are organic one, while insoluble solvents, they are not selected. Lipids which are extracted from samples, they depend on the nature of the organic solvents used. Now we'll discuss first method for estimation of lipids, and this is batch solvent extraction. In this method, what are the main steps? This involves mixing the sample and solvent in a funnel which is known as separating funnel. What is done here? Container is shaken vigorously so that organic and aqueous phases, they first of all they mix with each other and then they're allowed to separate. Aqueous phase is separated then, and the organic solvent is evaporated from the sample. When this organic phase is evaporated, what is left then? The lipid that is left. This lipid is measured. It is quantified with the help of some suitable balance, and its percentage is calculated. That okay, this sample contains this much lipid, and then we can convert it into its percentage. Next method is Slit extraction. In this method, lipid extraction from the sample is done by following method. Sample is first of all dried. It is ground into smaller pieces and placed in a porous thimble. And one can see here on the left side, the sample is placed in this small part. Thimble then is placed in an extraction chamber, and above this extraction chamber, that is placed in a flask which contains the solvent, and this sample containing chamber is below the condenser, which can be seen in the diagram. On heating the flask, the solvent evaporates, and when it evaporates, then it condenses back in the condenser, and it falls in the sample. In the sample, there is there may be lipid which gets dissolved in the solvent, and when it gets dissolved in the solvent, then it can be further measured. As the solvent passes through the sample, it extracts the lipids and carries them into the flask. The solvent is then evaporated, and the mass of the remaining lipid that is measured, and then it is used to calculate the percentage of the lipid in the sample. This procedure may be repeated many times so that we get better results, and this procedure is repeated from hours to days even. So it's quite a lengthy phase. Insoluble part of the sample that remains in the thimble, which is then discarded, and only one solvent is used again and again, This is its advantage. Instead of using many solvents at the same time, or instead of using many portions of the same solvent, one sample that is used again and again. So this is its advantage.